All right, so this is going to be the second lecture in photosynthesis. We'll be covering the light reactions. So if you remember, photosynthesis was divided up into two divisions. We have called the light reactions, and then we move on to the Calvin cycle. Now I want to point out, using the total formula for photosynthesis, what's happening generally in each stage. So for a formula for photosynthesis, we have carbon dioxide plus water is going to yield some glucose and some oxygen that's going to be given off. So in the light reactions, we do not use any carbon dioxide. We do, however, utilize water. And water is used by the photosystems in light reactions to supply hydrogen and an electron. Because in the light reactions, your primary goal is to absorb light energy with a pigment, use that energy from the pigment to excite or energize an electron. So now you can all of a sudden have chemical energy. And then that electron is utilized in those light reactions to help generate an energy-containing molecule like NADPH. Okay, we're going to use water, but we are going to make NADPH, and we're going to make a little bit of ATP, and we're going to give off some oxygen. So the oxygen that we're giving off is coming from the water. Really what water does is it supplies electrons and hydrogens to the light reactions. We'll look at that in just a minute. The Calvin cycle. The Calvin cycle uses your carbon dioxide, and it utilizes these energy-containing molecules, your NADPH and your ATP, and it utilizes those energy-containing molecules to assemble carbon dioxide and turn it into glucose. Now, keep in mind that when we say carbon dioxide, if this was a balanced chemical equation, I should have a coefficient of 6 in the front, because there are 6 carbons here in this one molecule of glucose. So what we're really doing is we have 6 individual carbon molecules here, 6 individual carbon dioxides. But by using the energy from these molecules here that we made in the light reactions, we can then assemble those 6 individual carbons and carbon dioxide and rearrange it into this carbohydrate or other carbohydrates. So that's what we want to ultimately do. So we're going to use carbon dioxide, we're going to use these energy-containing molecules, and we're going to make, ultimately, some glucose. Okay, so that's the overview. So let's get to the light reactions and what happens there. So obviously, if they're called the light reactions, you need light to make it work. So let's go ahead and make the thylakoid membrane, which is where this happens. Remember that the thylakoid were the membrane sacs inside of your chloroplast. Embedded in that membrane are your pigments, like chlorophyll. It's embedded in all throughout that membrane. So let's draw a bilayer here. So we've got our phospholipid bilayer. I'm going to make a green pigment. And another green pigment. Let's make some proteins that are here. We got some proteins. So a few things to remember as we go into this. So this is our lipid bilayer, right? It's selectively permeable. It gets to regulate what passes through and what does not pass through based upon the characteristics of the material. It can have proteins embedded in it, like these blue proteins. It can have these large proteins embedded, like these photosystems or these clusters of pigments that are stuck in it. Lots of things happen in a membrane. Also, membranes are really good at regulating or keeping things out, right, or separating materials. And that's what this is going to be used for. Not only does it support the structure of these pigments, these photosystems, but it also helps to separate some materials. So let's get to this. So first off, we need some light. So this whole time, you need to keep in mind that as long as light is shining, it's driving this whole photosystem. So let's just call this light. And let's say that this is the outside of the thylakoid, and this is the inside of the thylakoid. This is out, and this is in. Okay, now let's go ahead and let's energize, let's do what pigments do, and let's energize some electrons. But we have to get the electrons from somewhere to begin with. So the very first stage in this is you have to take some water, which is this water right here, which is when a plant uses water, this is what it needs it for. It takes this water, and at the very beginning of this, at what we call photosystem 2, let me just put photo 2. Photosystem 2 takes water and breaks it. And it breaks it into hydrogen. It breaks it into an electron, and it breaks it into some oxygen, right? Because H2O and so forth. Now the oxygen ultimately is just going to be given off. It's released. The plant cannot use that at this point. So if you go all the way here to the end of our formula for photosynthesis, this oxygen, the oxygen that plants always give off, everybody loves plants because they make oxygen. They're really making oxygen by splitting water and throwing away or releasing that excess oxygen. It can't use it for this process, so it releases it. So that's where your oxygen comes from, and we don't need it anymore in here. What we really want to get from this is this electron right here. Now this electron has not yet been energized, but that's what this photosystem wants to do, is to energize that electron. So what do? We energize it. So I'm going to put my electron in this photosystem. I'm going to say, hey, I'm all energized. I'm full of energy. Okay. Once that electron is energized using light energy, it starts to get passed along. You don't want to hang on to an energized electron. They're really energetic. It's like a hot potato. So it starts passing that along. It then moves through a series of proteins embedded in the membrane called an electron transport chain. So I'm going to move this right here. So I'm going to put my water and put my hydrogen and my electron and my O2 here. And then this section is called an electron transport chain, or the ETC. And the electron transport chain transports electrons. It moves electrons along a series of proteins. Here you want this, okay, I'll take it, okay, I'll pass it along. Now, while it's doing this, it's used for something. The energy in that electron is used for something. Remember, we still got this hydrogen down here that we haven't done anything with. And what happens is, as this electron, this energized electron, is passed along these proteins, it's used to drive a membrane pump, which transfers this hydrogen, which we still have just laying around here, through the membrane. So let me get a different color here. Let's use pink. And so while this electron's moving along, we move some hydrogen, some H pluses, some hydrogen ions, which is a proton. It moves it through the membrane. Now, we're going to get back to this in just a second. But every time you energize an electron, you're moving some hydrogen. So what's going to happen? This is active transport, right? And in active transport, we can move against diffusion, right? Active transport requires energy to move materials. You only need to do that if you want to pump it against the concentration gradient. So that means we're actually concentrating hydrogen on one side of this. It's really like putting water behind a dam. So we're building a bunch, can you see that? A little bit. We're building up a really high pile of hydrogen on this other side of this membrane. Not in here, but here, right? We're gonna get back to that in just a second. So we energize our electron, we shoot it down the electron transport chain. It uses its energy to move some hydrogen while it's moving along. But in doing so, it loses some of the energy it had, right? Because when you use energy, you can't get it back. So we have to re-energize that electron. That low energy electron is all worn out from pumping some hydrogen, gets back into what we call photosystem 1. Photosystem 1, and we have our electron there, and it becomes re-energized. Keep in mind that both of these happen subsequently, like at the same time, because light's hitting both of these. So it's like everything happens all at one time, and we're constantly splitting water, moving electrons, pumping hydrogen, re-energizing electrons all the time. As long as light is hitting a plant, and it has water and it has light, this is happening. Here, we re-energize that electron, we pass it along to another molecule, which then hooks it on and to this material called NADP, and NADP gets charged up and becomes NADPH. Right? So we said that NADP is a molecule that can hold on to, it's an electron accepting molecule. So that little electron joins in on this, and we grab a hydrogen and we form NADPH, which is energized, and that was the material we said we were going to send over to the Calvin cycle. So this is one of the end products, NADPH, right? This is holding our energy. Now, unlike just a free electron sitting around, it can hold on to it for a bit longer, and we can move it. It's those oven mitts that can grab the electron and move it around inside of this chloroplast instead of just letting it float around. Okay, so a couple questions here. Ultimately, we wanted to get our electron hooked up in here. It's good, we've got it. But we haven't answered a couple other questions. Like, one, I told you earlier, we make some ATP. Where does that get made? And two, what's all this hydrogen going to be used for? So let's kind of, I'm going to erase this, and we'll back up, and we'll look at another section of the membrane. So let's get rid of our light, and let's just erase this center part here. All right, so this is the same thylakoid membrane. This all happens in the same kind of place. Just for us to be able to see it all right now, I'm going to draw it out. So we've still got our lipid bilayer. And we said that in our lipid bilayer, we had a bunch of hydrogen on one side. Remember, we pumped it across in the electron transport chain. Now, if you've got a lot of hydrogen up here and not a lot of hydrogen down there, what wants to happen? You want to move from high to low, right? This is high, this is low. And we know that in diffusion, you always want to move from high to low, right? But you can't, because this lipid bilayer is selectively permeable. It will not let charged particles through it. Hydrogen cannot go through this unless somebody helps it get through, okay? So there is a molecule embedded in this called ATP synthase. Now I'm just drawing it as a little like big block or something. It's not quite what it looks like. It's a really interesting protein. And we want to talk about what ATP synthase does. So this is embedded in the membrane. Earlier on, we put all our hydrogens up here. There's not many hydrogen down here. Here's high, here's low. The hydrogens want to go through here. Well, we had talked in the previous chapters about how proteins and enzymes can act as, help move things through the membrane. This molecule, ATP synthase, ATP synthase provides a channel to let the hydrogens pass through the membrane from high to low. What makes it go from high to low? Diffusion, right? They're getting pushed through from the force of diffusion. The really cool thing about ATP synthase is it acts like a biological turbine. As this gets forced through, it allows this protein to flex and helps a chemical reaction occur. Now this looks really similar to something you're familiar with in real life. This is like the dam. This is the water behind the dam, and this is outside the dam. You've all seen probably water being held back by a dam. The water goes through the dam, and as it goes through, it can be utilized because it's pushing. It can be utilized to generate some sort of energy. In this case, it's a hydroelectric dam, and it generates electricity. This is really similar, but instead of generating electricity, it allows a chemical reaction to happen. It allows it to form a bond. So this hydrogen provides the energy as it's falling down its concentration gradient, just like water moving down can power things, like dams and mills in the old times and so forth. And as this hydrogen goes through, it is used to convert ATP, adenosine diphosphate, which is not charged up. It can hook on a third phosphate and form adenosine triphosphate, which has energy in it. So this is, okay, now I love this. A lot of molecules that are enzymes are named for what they do. So ATP synthase, this stuff, what does it do? It synthesizes ATP. That's great. I love it when we finally name something with a name describing exactly what it does. All ATP synthase does is makes ATP. And it does that by simply hooking on that third phosphate back on to that adenosine diphosphate and turns it back into triphosphate. It stores energy in that bond. It gets the energy from the falling hydrogen. Okay? So now, at the end of this, as long as we're running our light reactions, our photosystems, and our electron transport chain, not only are we generating some energized electrons in NADPH, but we're also maintaining this concentration gradient and we're generating some ATP. So we get both ATP and NADPH being produced. Both of those are holding energy. We're going to use those in the light reactions to hook together our sugar up here. We're not there yet, and we'll look at that next time. Keep in mind, the only thing we've used, though, so far is this hydrogen. We haven't touched our carbon, and we haven't made any sugar, but we did make our oxygen. Our oxygen is a byproduct of this process.